Buddy. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. We're here at Space Place. Thank you again, Kay Creewell, for hosting us. This is the start of, I think, the 17th year, maybe 18th. I can't remember. We'll have to go back. I think Paul Brandel says it started the same time as Wednesday Night the Lab, and we're starting the 18th year of Wednesday Night the Lab today. So thank you. We usually go around the first day, but since the weather isn't so great, we'll probably try that next week. Next week, Susan and Steve Carpenter will be here to talk about their work with prairie restoration and other land restoration in the driftless area. So that'll be 1 o'clock next week, the 1st of March, here at Space Place or online, 2 ways, through the Zoom or through the live feed to the Wednesday Night at the Lab YouTube site. All right, today it's my pleasure to introduce to you Paige Henning. She's a NSF funded postdoc, which means she went out and wrote the grant to get it, which is a very hard thing to do. She works at the Genomics Center. She's a botanist. Yes. Look, there's 2. Only she knows what she's doing. Paige, I'm going to ask you the 5 questions I ask everybody. You can answer in any way that you'd like. Standard line is the answers don't have to be true. They just have to be believable. Paige, where were you born?
Spokane, Washington.
Well spoken. And where'd you go to high school?
North Central High School in Spokane, Washington.
All right.
Yeah.
And then where'd you go for your undergrad and what did you study?
So I did 2 years at community college where I got an associate's in biology, and then I did 3 years at Washington State University where I got my bachelor's in genetics and cell bio.
There you go.
Yep.
And where is Washington State University?
In Pullman, Washington.
And where is Pullman, Washington?
It's south of Spokane, about an hour and a half south.
Yep. Right on the border with the great state of
Idaho.
Let's call it Idaho. Okay. It always amazes me that Washington State and the University of Idaho are about 7.3 million miles apart. So it's the 2 closest land grant universities. And where'd you go for your PhD?
Also Washington State University. And I got my PhD in just biology, but I worked on plant reproduction.
Excellent. And then when did you arrive here?
In June of 2022.
Right. That was a good month to arrive. For people that are arriving in January and February like I did. Today, you're going to talk about heteromorphic self-incompatibility, a peculiar case of convergent evolution. Would you please join me in welcoming Paige Henning to PLATO?
Well, now that you know my life story, I guess I'd like to start off by telling you that I love heteromorphic self-incompatibility. It's what I worked on for my PhD, and I've been lucky enough to be funded and get to continue my project in the Sussman Lab at the University of Wisconsin-Madison. So I'm going to start off with a controversial statement, which some people may be upset about. Charles Darwin was a botanist, and other botanists will agree, or plant biologists. My evidence here is that during the later years of his life, the last 20 years of his life, approximately, he actually published 6 different books on plants. And in his autobiography, he is quoted saying, "I do not think anything in my scientific life has given me so much satisfaction as making out the meaning of structures in these plants." Charles Darwin was particularly interested in the flower. So the flower is probably the thing everyone is interested in when it comes to plants, or maybe the food itself. So to begin, let's talk about the evolution of the angiosperm and the evolution of the flower. So the flower actually is considered what has made the angiosperm such a successful group of plants. So the last common ancestor of the angiosperm was about 136 to 141 million years ago. You'd imagine that's a rather short time for a species, for a group of species to actually essentially conquer the earth. So 79% of the known plants are actually angiosperms, and they are the only plant group that actually is represented on every continent. So there are grasses in Antarctica, and those grasses are angiosperm because grasses are angiosperm. The majority of plants that you think of are probably angiosperm. So for example, we have hazelnut. There's the beautiful flowers of the hazelnut tree. We have your hedges, which you'd see in like mazes, I guess. Beautiful white flowers. And then your grasses are actually also angiosperm. And there's a representation of a grass flower, like your yard grass. Beyond that, angiosperms are also very important in terms of like food, right? Basically, any vegetative food that you can think of is probably going to be produced by an angiosperm. So the flowers are probably the flower is probably what contributed most to the success of the angiosperm. So let's talk about flower morphology, because I realize that not everyone is a plant biologist and we might need some refreshers, or maybe I can educate you for the first time on floral morphology. So here is a cartoon diagram of a flower, and next to it is a actual representative flower of Turner subulata. So here we have our stamen. This is our male reproductive organ. We can cut our stamen up into two different sections. We have our anther. This is where our pollen is going to be produced. Our pollen is going to contain the sperm. And then we have our filament. Our filament is a stocky structure that's going to hold the anther in place within our flower. Here we have the pistil. Our pistil is our female reproductive organ. This can be categorized into our stigma. So our stigma is where our pollen is going to land and begin to germinate. We then have our style. Our pollen tube is going to elongate through the style and eventually reach the ovary, which is not diagrammed here, but would be somewhere in this area. I bring up these terminologies because I'm going to be using them throughout the talk, and it's best that we're all on the same wavelength. So I'm interested in self-incompatibility. So what is self-compatible versus self-incompatible? So a self-compatible individual is going to have bisexual flowers, is going to be capable of reproducing with itself, as well as reproducing with other individuals within a population. In self-incompatible individuals, we're incapable of reproducing with ourselves, but we are capable of reproducing with other individuals within our population. So you can kind of think of this like sex with animals. You're incapable of reproducing with yourself, at least most animals are. When we talk about self-incompatibility, we use the term mating type to describe who a plant is capable of reproducing with. With self-incompatibility, we have two different flavors. We have our homomorphic variant, which is where we only have one floral variant. These are genetically very complex. We have a single locus, but we're going to have 60 or more alleles typically. Our classic example of this would be in Brassica. And then we have heteromorphic self-incompatibility, which is the focus of today's talk. We're going to have two or more floral variants, and we're going to refer to these as morphs. And again, we can think about these as like sexes with animals. So we have our S-morph individual who's only capable of reproducing with an L-morph, and vice versa. Genetically, these are relatively simple. It's hypothesized that it's going to be controlled by one or two loci. And we have some examples. So primrose or primula is known for distyly, and then we see tristyly and oxalis. And I'm going to explain distyly and tristyly more after I go on about Darwin some more. So like I said, Darwin wrote 6 different books just focused on plants in general. Now, the book that I'm the most interested in, and the only one I bothered to read because I'm terrible, is his 1877 publication, The Different Forms of Flowers on Plants of the Same Species. Within this book, there are 8 chapters, and if you were to just look at the chapter titles and not actually bother to read them, you would notice that 5 of them are about heterostyly. If you did read them, you would know that 6 of them are about heterostyly. So Darwin was pretty interested in heterostyly. So like I said, before I get into what he actually talked about in this book, heterostyly, we have 2 different floral morphs. This is a plant reproductive system that's going to confer self-incompatibility and intramorph incompatibility. So we have our distyly, which contains 2 morphs that differ by the placement of their pistil versus their anther. So our S-morph individual is only going to be capable of reproducing with an L-morph and vice versa. They're named after the length of their style. So we have our short style individual or a short morph and our long morph. Distyle is the most popular form of heterostyle. So there's at least 232 genera that have distylous species, if not more. Then we have tristyle. It's very rare. There are 5 genera that only have tristylous species, meaning there's no distylous species within that genre. And there's 15 genera in general with tristylous species. Here we see 3 different morphs. The reproduction is much more complex, and we're just going to ignore it because we love distyle. Yes. No. So just like on one human, you can be a female or a male. Biologically, on one plant, you can be an L or an S. That's why I like to refer to it as sexes, because I feel like that's an easier way for everyone to conceptualize what it means to be S-morph versus L-morph. Yeah. Cool. Thank you for the question. I probably should have repeated it. Okay, so back to Darwin's book. So this book was composed of a series of garden experiments where he did a series of crosses between the L-morph and S-morph individual of Primula elatora, Sensinus, Varus, and Vulgaris. So this is just a diagram from his book, again, exhibiting the reciprocal orchogamy, or that distance between the stamen and the stigma. This distance was already observed prior. I mean, the first time that distyle was documented in Primula was way back in the 1500s. And it was common for just horticulturalists to like know the morphs. They were named by horticulturalists to begin with. Yeah. What Darwin did during these crosses is he performed a series of crosses, legitimate crosses, where we would have an L-morph individual mating with an S-morph individual, and illegitimate crosses, where we would either have an S-morph individual mating with itself or an S-morph individual mating with a different S-morph. Same with the L-morph. So we'd have an L-morph individual mating with itself, or an L-morph individual mating with a different L-morph individual. So what he found is that there are differences in reproductive success during illegitimate crosses. So if an L-morph individual mates with itself, it's actually going to produce more seed than if an S-morph individual mates with itself. However, legitimate crosses always produce more seed than illegitimate crosses, suggesting that this system exists to promote outcrossing. It exists to promote the L-morph to breed with the S-morph, and vice versa. He also observed differences in seed weight. So the S-morph individual actually has heavier seeds. He noticed actual differences in degrees of reciprocal orchogamy. So the distance between the stamen of the long morph and the stigma of the L-morph is greater than that of the S-morph. He was the first person to actually measure this out, I guess. He noticed differences in pollen grain size. So this had been documented once before in Primula vulgaris, but he expanded on this and showed that it was a common trait across distylus primula. He noticed differences in stigma texture. And he noticed differences in natural occurrence of self-pollination. So for this, he took a bristled brush and he shoved the bristled brush down each flower. He looked at where the pollen landed on the bristles brushed, and then also looked at the stigma afterwards to see if pollen from self had landed on the stigma. He noticed that pollen from the L-morph has a very high rate of landing on the L-morph stigma, and this is likely due to the anther being placed below the stigma. As the brush would be pulled out, the pollen would fall onto the stigma, and you would not observe that with the S-morph. So, Charles Darwin, he was also a fortune teller, I guess, because from his autobiography, he talks about how he actually did love heterostyle, but nobody would really notice any results related to heterostyle, and it would be essentially a very unknown topic. And as you will notice from today's talk, it's a very small research group that's working on distyle. But I guess I'll read this. As before remarked, no little discovery of mine has ever given me so much pleasure as making out the meaning of heterostyled flowers. The results of crossing such flowers in an illegitimate manner, I believe to be very important as bearing on sterility of hybrids, although these results have not been noticed, but have been noticed by only a few persons. So After I read that, I kind of cried a little bit because I understood how Darwin felt. Please laugh at my jokes. Okay, thank you. Hopefully everyone at home is laughing too. So what are the characteristics of a distylus plant? So if we're going to go around claiming everything is distylus or not distylus, we need to set some general ground rules. So as already previously stated, we have differences in our pistil length and our anther height. And the idea behind this is that it will promote pollen to land on the region of the pollinator that will then be touched by the opposite stigma. So pollen from the anther of the S-morph should land lower on the pollinator, which will then be at the same region of the stigma of the L-morph and vice versa. So you would imagine if this little bee goes into this flower, it's going to get pollen down there. When he goes into the next flower, that pollen should fall onto the stigma of the opposite morph. We have self-incompatibility and intramorph incompatibility, as I've spoken on. And what Darwin couldn't see because of the lack of electron microscopy at the time is not only are the pollen grains, they don't only differ in size, but they also differ in their pollen coat. So here I have stolen a picture of linum. In A, we can see the pollen coat of a short morph individual of linum versus the pollen coat of an L-morph individual of linum. And you'll notice there are textural differences between the two morphs pollen. We also see with their stigmatic papial differences in the texture. So here is the stigma of a S-morph individual and that of an L-morph individual. It's hypothesized that these differences in the stigma and the pollen exist to promote the pollen of the opposite morph to stick to the stigma while preventing the pollen of the same morph from sticking to the stigma. So I talked about how this is a case of convergent evolution, and now I'm going to show you why. I also didn't anticipate for these little things to be on top of my screen, so I apologize for that. So heterostyly, as I stated before, it's evolved a bunch of times, right? It's evolved independently in at least 28 different families and at least 237 genera. I've found throughout studying that some genre, it seems like they probably haven't been documented with heterostyly, which is actually something that other people have noted, which is why botany is important, I suppose. It's found in both monocots. So these three families up here are monocots and dicots. Yeah. So how is distyly occurring, I guess? What's the genetic basis? That's what I was interested in for my PhD. So throughout time, there have been a series of hypothetical genetic bases for distyly. Darwin himself never commented on a genetic basis, and that's because Darwin did not know what, he didn't have Mendel's work to go off of at that point, right? Yeah, I'm sorry, I had a pause in my brain. Okay, so in 1905, Bateson and Gregory would actually determine that the S-morph is likely heterozygous for the distyly gene, and the L-morph is homozygous. They determined this by basically crossing the two individuals and then counting the number of offspring of each morph. They found that there's always a 1-to-1 ratio, which suggests that the S-morph has to be heterozygous and that homozygous dominant individuals must be lethal. So we're at this story where we have heterozygosity versus homozygous recessive. In 1928, Ernest would actually determine that there are likely two distyly genes, which he would now call the S genes. He would call these genes gene G and gene A. He hypothesized that G was involved in all female aspects of distyly, while A was involved in all male aspects of distyly. And he did this by looking at a homostyle mutant, so a mutant that is no longer distylous. In 1933, he would come back and overturn his own hypothesis, and he would determine that there are at least 3 S genes. He would now name these GPA. So G would still be the gene that's involved in all female aspects of distyly, but now after observing a new mutant, he would determine that P has to be a gene related to just male mating type determination, and A has to be a gene that determines male morphology. In 1950, mathers, he did some math, and I'm not going to explain the math because I don't really understand it, but he determined that the S genes are likely part of a supergene. So supergenes are really cool because this is multiple genes that are always inherited as a single Mendelian unit, which is why we are capable of always getting a 1-to-1 ratio of the S morphs, despite there hypothetically being 3 genes. And he would call this supergene the S locus. And in 1956, Pamela and Dowick, they would determine that there are actually probably 5 S genes. The female morphology gene, a female mating type gene, a pollen size dimorphism gene, a male mating type gene, and a male morphology gene. And that was a lot. There have been new hypotheses since, throwing up to like, I think 12 different S genes, but we're not going to talk about those hypotheses because these are this is the classical like historical viewpoint of distyly. So before we actually get into all the cool science, I want to just put some pictures to names and also scientific nomenclature, because if I tell you Phagopyrum esculentum, you probably don't know that that's buckwheat. So some distylate species, we have Phagopyrum, which is our common buckwheat. We have a series of different primulas that I'll talk about, and I've already talked about, which is primrose. We have Linum tenor. Linum is part of the same family as flax. We have Turnera, which is a family of subtropical shrubs from South America, Central America, as well as Africa. This is my species of interest, and they're closely related to passion flower, which is why they are beautiful. And finally, we have Glycelium eleganus, which is the heartbreak grass. It's closely related to primrose. So in 2012, the first S gene would actually be identified. 2012 was actually a very good year for distyly because 3 different S genes would be identified, but I'm only going to talk to you about the Phagopyrum one. So Yasu's group in 2012 would publish a paper where they performed RNA-seq of the 2 morphs of Phagopyrum. They would then identify genes that were only expressed in the S morph and not the L morph. They then went through and took a list of these genes and genotyped the L morph and the S morph individual for these genes. The one gene that they found that was specific to the S morph was this S locus early flowering 3 gene. It is a homolog of the Arabidopsis transcription factor L3, which is involved in regulating flowering time, circadian rhythm, and even is involved in repressing elongation. This suggests that it has the potential to be the female morphology gene, seeing that it's only expressed in that short style morph, and thus is likely repressing elongation of the style. So RNA-seq is when you take all of the RNA out of a tissue, and then you convert it into DNA using a enzyme called reverse transcriptase, and then you send it off to be sequenced just like you would DNA. Yeah, no problem. So once they had this gene and they showed that it was present in the S morph of one ecotype of Phagopyrum, they went and they genotyped a bunch of different individuals from a bunch of different ecotypes, and they found across the board that self3 was only in the S morph's genome. Furthermore, they did look at a homozygous mutant, which was a long style, long filament mutant, and they did find that self3 was missing, which supported the hypothesis that it is likely the female morphology gene. So that was pretty exciting. Oh, I forgot to show you this. Okay, so they also looked at 3 different distylus species of Phagopyrum, and they found that all 3 of them had this gene in that homozygous, in that homostylus mutant. So the mutant, they found that there was actually a mutation, a nonsense mutation, so an inappropriate stop codon. And then in one species that does not have distyly, they found that there was also a mutation within this gene, likely what is causing it to no longer be distylous. Sorry. So in 2016, we would have the first characterized S locus. I skipped the other two S genes because we're going to cover them while we talk about S loci. So the first characterized S locus was in Primula, Primula vulgaris. So what they did is they sequenced the genome of the S morph as well as the genome of the L morph. And they looked for regions that were specific to the S morph and non-existent in the L morph. They found a hemizygous region that contained 5 genes. So this is our S morph individual. It contains these 5 genes that our L morph individual does not contain. And you'll notice that there is not an allelic counterpart here. So when I think of hemizygosity, I like to kind of think of the Y chromosome because you only have one copy of the Y chromosome if you are a biological male. This is not a chromosome by itself. It's just a region of the chromosome, just a super gene, right? But it only has one copy. The pen has nothing here. Or the pen, sorry, the L morph. To further support that this region was actually involved in the occurrence of distyly, and also to try and tease apart what these genes might be involved in terms of distyly, they looked at 2 different mutants. So here they have a long homostyle mutant that is capable of reproducing with itself. They found a mutation in the CYP in 2 different individuals, and so they determined that CYP is likely the female morphology gene. Similar, they had a short homostyle mutant, and here this is a self-incompatible individual still, but they find a mutation in this GLO-T, which suggests that GLO-T is likely involved in male morphology. They were unsure what the remaining genes are involved in, and to this day, they still don't know what those 3 genes are involved in, but they know what CYP and GLO are involved in. Okay. So in 2019, the second S locus would be identified in a project that I was involved in. So here, similar to the Primula group, we also had a long homostyle and a short homostyle mutant. We did a series of back clones to identify regions of the genome that were likely S morph specific. Once we had identified 3 putative genes for the S locus, so SPH1, YUC6, and BAD, we genotyped for those genes in 5 different species of Turnera, and we found that these genes are all S morph specific. So the S's represent S morph individuals, and the L's represent L morph individuals. And you can see the S morph individuals all genotype for these 3 genes, while the L morph do not. So that supported that these are likely the S genes per Turnera. When we looked for these genes within our L morph individual, or our long homostyle individual that is self-compatible, we found a mutation in BAD, which would cause truncation, and thus we determined that BAD is likely the female morphology gene. When we looked for mutations within our short homostyle, we didn't find any within our S morph within our S genes, but we did find that SPH is not expressed in the short homostyle, which suggests that SPH is the male morphology gene by the process of elimination that would leave YUC as our male mating type gene. So we had these genes, and now we are interested in, is our S locus hemizygous like the Primula S locus? And it is. So we used our series of back clones to build a scaffold of where the S locus was within our S morph individual and where it should be in our L morph individual. And we find that, again, we have hemizygosity of the S locus within Turnera. We also found that there are these inversions that occur. So if you were to look at the chromosome that contains the S locus, we have, for example, the gene order of RBP, ERF, and CBSX versus the homologous chromosome that lacks that S locus. We have the gene order now of CBSX, ERF, and RBP. And this occurs at both borders of the S locus. This likely this inversion likely represses recombination at the S locus, right? So that's likely what's keeping the S locus together as a supergene and preventing recombination from occurring. So once we have our S genes and we know we have our hypotheses regarding what they're doing in terms of distyly, we could use Arabidopsis homologs to come up with hypotheses as to how they are influencing these various aspects of distyly. So like I said, SPH1, we predict to be our male morphology gene. And based off of homology with Arabidopsis and Poppy, we think that is a signaling peptide. YUC6, we hypothesize as our male mating type gene, is a homolog of YUC6 and Arabidopsis, which is involved in auxin production. Auxin is a plant hormone. And then we have BAD, which is our female morphology and mating type gene. It is a homolog of a BAD family member, BRI1, in Arabidopsis, which is involved in inactivation of rasosteroids. Rasosteroids are a signaling molecule. They've been classified kind of as a pseudo hormone because originally they were thought to be a hormone, but they are not a hormone. To be a hormone, you have to leave the tissue that you were synthesized in, or the cell that you were synthesized in. One or the other. Okay, so once we had identified our genes and we have our hypothesized modes of actions, we were able to come up with a model for how self-incompatibility is likely established within Ternera. So, as I said before, BAD is likely expressed in our pistil, SPH in our filament, and YUC in our stamen. Based off of the hypothesized role of YUC6, we can say that our stamen likely has a high auxin environment, and so pollen is likely being developed in a high auxin environment. Versus the pollen of the L-morph individual, which is likely being developed in a low auxin environment relative to that of the S-morph. Vice versa, because BAD is likely involved in rasosteroid inactivation, we can say that the stigma of the S-morph individual is likely a low rasosteroid environment versus that of the L-morph, which is likely a high rasosteroid environment relative to the S-morph individual. So we basically have a hypothesis that our mode of self-incompatibility is just a high matches with high, low matches with low. So pollen that's developed in a high auxin environment is only capable of germinating on a stigma that has a high rasosteroid environment, and vice versa. We would actually later overturn this hypothesis, but I'm not going to talk about that. So yeah. So this was the first model for how self-incompatibility might be established within a distylus species. So within last year, which was a pretty cool year for distyly, 2 other S loci were sequenced and published. So the genome of linum was published in 2022. They identified another hemizygous region. This region contains 8 different genes. So TSS1 was actually identified back in 2012 after the pegopyrum gene. It was never genotyped before, so it wasn't, nobody was sure if it really was an S gene, but it was a gene that was only expressed in the style. So this actually confirmed its presence as an S gene. Interestingly, they found that one gene was allelic. So this little gene right here that they didn't bother to name, and I'm not going to just read those numbers off for you, is actually truncated in the L-morph individual. So it has it doesn't have a stop codon. Interestingly, though, while there are 9 genes, only 5 are expressed. So it's unclear what those other 4 are doing. It's actually unclear what almost all of them are doing, except for TSS1, which they think is involved in stigma repression, and WDR, which they think is involved in male morphology. So in 2022, the S loci of Gelasium would also be published. It contains 4 genes, 3 are hemizygous, and 1 that is allelic. Similar to the linum story, the 1 allelic gene is truncated. What's interesting is just Gelselium is closely related to primrose, and we actually see that CYP, which is the female morphology mating type gene in primrose, primula, is also present in the S locus of Gelselium. Interestingly, though, it appears that they both underwent independent duplication events from the same ancestral gene, so that was cool. So I blabbered on about this just to tell you that convergence also occurs at the genomic level, right? So it appears as time goes on that hemizygosity is required for distyly to occur, or at least so far from what we have seen, which is pretty cool. So the next question is, how do we evolve distyly? So back in the 70s and also in the 90s, two different models for the evolution of distyly were published. So Charlesworth and Charlesworth in the 70s published the self-avoidance model, where inbreeding depression promotes the plant to evolve some form of self-incompatibility. Then to prevent male gamete wastage, so to prevent the pollen from self landing on the stigma of self, you would evolve reciprocal erkogamy, eventually resulting in the distylus plant. Webb and Lloyd in the 90s would suggest this pollen transfer model as a different way to evolve distyly. So we would start off with a plant that is approaching herkogamy. So there are some differences in style length and filament length, but we have not reached true herkogamy yet. We would then continue to move until we get to reciprocal erkogamy, and eventually we would evolve self-incompatibility again to prevent that male gamete wastage, likely in the L-morph individual. So to predict how distyly would evolve within a species, we would need to predict the order by which the S genes were recruited to the S locus, which we can do using synonymous mutation rates. So this is when we look for changes at the nucleotide level that do not change the amino acid sequence of the protein. Beyond just evolving distyly, how do we build a supergene? So Keppel back in 2017 proposed two different hypotheses for how one could evolve a supergene. So we could use a step-by-step duplication method by which each supergene is independently duplicated into the S locus until we eventually have our finalized S locus. Alternatively, we could have a segmental duplication where all of the S genes are then duplicated into what would later become the S locus. You could also have like some varieties where like two genes are duplicated and then one gene is like independent, you know, whatever flavor you want, but this is the general concept. So to figure out how your S supergene, your S locus was built, you would need to identify the genomic position of all the S genes homologs, which is what they did in Primula back in 2022. So after sequencing the genome of Primula varus and actually getting to the chromosomal level, they identified the homologs for all five of Primula's S genes within two different species. And they found that the S locus likely evolved by a stepwise situation in Primula because each of their homologs was on a different chromosome rather than neighboring one another in a different chromosome, a different genome. They looked at synonymous mutation rates, and they were actually able to determine that this KFB was the first S gene that would then be followed by CYP. Unfortunately, because we don't know what KFB is doing in terms of distyly, there's no way to predict whether or not this follows the Charlesworth model or the Lloyd-Webb model of evolution. What was also rather interesting is they noticed that bordering the S locus, there are 4 different CFBs. So this gene CFB, there are 4 copies of it bordering the S locus, 2 on either side. They all seem to have duplicated from one progenitor. So they were interested in this. How did this evolve and why did it evolve? So we imagine that originally there was one CFB, which you would have to imagine is there on both sides. and a duplication occurred, a tandem duplication. Our S locus was then acquired via each stepwise duplication events. And then we have a second segmental duplication where these CFBs are duplicated to the opposite border of the S locus. Thanks to non-homologous recombination of these CFBs, we would hypothetically lose our S locus on one of the chromosomes, which would ultimately lead to the hemizygosity of the S locus. So this is pretty cool because it's the only model for which model for how a dicellus species has evolved hemizygosity of the S locus. Yeah. So with the sequencing of the Ternera genome, we were capable of also determining that the S locus in Ternera evolved via a stepwise duplication event. I don't have any pretty graphs to show of the stepwise duplication for Ternera, but I do have a very cool image to show you of the bordering regions of the S locus. So interestingly, unlike Primula, the regions that bordered the S locus evolved prior to the S locus. So if we look all the way back at grapes, ovitis, and populus, we can actually see that the bordering regions are there within the genome and have not undergone an inversion or anything. So this means that the inversion is specific to Ternera, but the region itself is not, unlike Primula. We also looked at synonymous substitution rates, and we were able to determine that YUC6 is potentially the first S gene. We would need to expand our analysis to determine if this is true or not, potentially suggesting that Ternera evolved via that self-avoidance model or that Charles-Worth model. Beyond this, we showed that the S locus evolved within Ternera itself. So we compared, I used a phylogenetic analysis to identify the closest homologue of the 3 S genes within Ternera across, it was like 70 different species of angiosperm. And I found that each gene likely evolved within Ternera itself. So with the publication of their S loci, there was also the publication of how these S genes evolved. So in Linum, again, we see that stepwise duplication. When they looked at homologs within their genome, they found that all 5 of the genes reside on different chromosomes than the other S genes, likely suggesting that they evolved via stepwise duplication events. This was further supported by synonymous substitution rates. So they showed that WDR40 likely evolved prior to these other 2 genes. They did not look at the synonymous substitution rates of all 5 genes, so we're unsure which gene evolved first. In Gelsilium, the S locus likely evolved by a segmental duplication event. So when they looked at another closely related species, they found that the S locus was actually intact within Okay, that was just switched off. That was just switched off, it's okay. Yeah. Okay, so the S locus likely evolved by a segmental duplication event. So that was pretty cool because it suggests that S loci, they're capable of evolving either via these stepwise manners or by complete segmental duplications. So once we have all these S genes, now it's time to actually characterize them so that we can actually provide empirical evidence for their hypothesized roles. So in 2016, Hughes group would treat floral buds of Primula with brassilinoid. Brassilinoid is just a derivative of breast and steroids. When they did this, so these are our wild type individuals. We have our L morph and our S morph. And here we have a naturally occurring homostyle mutant. When they apply brassilinoid to the developing buds, we see that there's not really that much of a difference in our L morph individual. Statistically, there is a bit of a difference, but you obviously can see that there is a very large difference in the length of the style in the S morph individual to the point where it looks like a long homostyle at this point. when they looked at the cells of the the wild type individuals and lures that were treated with bracillinoid, they found that in the S-morph, we have much smaller cells in the style than we see in the L-morph individual. And when we treat our buds with this bracillinoid, we see elongation of those cells within the S-morph, which suggests that CYP is inactivating bracillinoid, and this is preventing cell elongation, which then prevents elongation of the pistil. The next thing they did in 2021 was that they actually treated the buds with the bracillinoid again, but instead now they did a series of crosses to see if CYP was involved in female mating type or not. So here we can see our two wild type crosses. When we cross our L-morph individual with an S-morph, we get approximately like 150 seeds, sure. And we get the same when we cross an S-morph individual with an L-morph individual. The first, this being like acting as the father, and this is the mother. When the L-morph acts as the pollen donor in a cross with a steroid or a bracillinoid treated S-morph individual, we actually see a reduction in seed set, which suggests that potentially the stigma and the entire pistil of this S-morph individual is now being recognized as an L-morph individual. Interestingly, however, when we use the S-morph individual, a wild type S-morph individual, as a pollen donor with this steroid treated S-morph individual, we see an increase in seed set that we would not see normally, which again supports this hypothesis that the S-morph, the steroid treated S-morph individual is now being perceived as an L-morph individual. When we these individuals, S-morph when it is self produces basically no seed. L-morph produces some, but when we the steroid treated individual, we see this massive increase in seed production, again suggesting that the stigma is now being perceived as an L-morph individual, while the pollen itself is still being perceived as S-morph pollen. So that is my long-winded way of saying that CYP is involved in both female morphology and female mating type. Interestingly, this might suggest that steroids are what's preventing pollen tube elongation within when a L-morph individual is selfed. However, when they treated elongating pollen with steroids, they did not see a negative response in the L-morph individual, suggesting that steroids alone are not the basis of female mating type, but that they are involved somehow. We just don't know how yet. Pollen tubes, yeah. So, yeah, so like the idea is when self-pollen lands on self-stigma, the pollen tube starts to grow through the style, but then it aborts randomly, right? And so the idea is if you treat the L-morph's pollen with steroid, since the L-morph's pistil should have a higher steroid level than that of the S-morph, you should see abortion of the L-morph's tube. However, we saw an increase in elongation, or Hughes group saw an increase in elongation, suggesting that it's actually having a positive effect on it. Slight positive effect, but positive effect, which is odd. So we've now characterized the female morphology and female mating type gene in Primula. So now we're going to do it in Turnera. So in 2021, a master's student in the lab that I worked in published this paper where she ectopically expressed BAD in Arabidopsis, and this produced dwarf plants. So here we see the wild type Arabidopsis, which is big, beautiful, and then we see our little tiny dwarf Arabidopsis that are expressing that TS BAD. To determine if BAD is potentially involved in steroid inactivation, she also performed qPCR. So this is when we're looking at RNA for a series of steroid related genes in Arabidopsis, so DOR4, CBD, and BAS1. And she found she's found that these genes were differentially expressed in a response to the expression of BAD within the Arabidopsis. So this supports the role of BAD, at the very least, in female morphology. As remember, we have that short style in that plant. And we have short plants in response to just ectopic expression. So next, we had a mutant copy of BAD from a different member of Ternera. So this mutant Ternera is self-compatible and also is a long homostyle. When in 2022, when Madsky expressed this mutant copy in Arabidopsis, we did not actually see a door phenotype. So here's our wild type Arabidopsis on the far right. And then next to our wild type Arabidopsis, we have Arabidopsis that's expressing that mutant BAD compared to Arabidopsis that's expressing the wild type BAD. So this supports that BAD is likely also the female mating type gene, as well as the female morphology gene. So I treated pollen tubes with two different derivatives of racestosteroids. So when I treated pollen tubes with bracillinoid under the same hypothesis as the primrose people, except for I published this prior to the primrose, before they did. Okay, so so we treated elongating pollen tubes with different derivatives of racestosteroids. When I treat the S-morph individuals with bracillinoid, we actually see an increase in pollen tube elongation, and we do not see any influence on the L-morph individual's pollen tube, which suggests that bracillinoid's not influencing elongation within the L-morph at all, but is positively influencing the S-morph's pollen, which was cool and like not exactly what we anticipated, but was cool regardless. And then we were confused because when you treat the pollen tubes with castasterone, another racestosteroid derivative, we actually see that there is a significant pollen or positive influence on the L-morph's tubes, which suggests that this is a story similar to that of the primula story, where racestosteroid alone cannot explain female mating type. There's something deeper there. So back to the convergent evolution aspect, by just characterizing BAD and CYP, we were showing that convergence occurs at a genetic level as well as a metabolic level, because we are likely seeing the same genetic pathways, just different genes being involved in style length dimorphisms as well as female mating type. And this is likely in both species involving racestosteroids somehow, which was pretty cool. This convergence at genetic level would later be expanded on in a 2020 paper that I published, but that's all computational, so I decided to ignore. So next, Leonard's lab characterized the primula GLO2 gene. So GLO2, they silenced GLO2 using virus-induced silencing. So they decreased expression of GLO2. And by doing so, they made short homostyle mutants. So here's our wild type S-morph individual, and you can see that the stamen is high up in the corolla versus in our virus-induced silence individual, our stamen are now low in the corolla, similar to that of the L-morph individuals, who you can see again, their stamen is approximately at the same height, and it doesn't really change when they try to induce silencing. Okay, and so they hypothesize that this is likely due to decrease in cell proliferation below the stamen itself. So that was pretty cool. That was a little tiny experiment in a very big paper. And then I characterized Ternera's YUC6 gene. So this was the hypothesized male mating type gene. So I used antisense, an antisense fragment. So this is just part of YUC6 that's been inverted on the 35S promoter to knock down expression of YUC6 in Ternera joellii. Ternera joellii is an obligate outcrosser, meaning that when you self Ternera Jolia, it never produces seed. However, when I knock down YUC6 in these S-morph individuals, we do see seed set when crossed with itself, and we see seed set when these mutant individuals are crossed with the S-morph. So this should not happen. This suggests that the pollen is being perceived as if it is L-morph pollen, despite it coming from an S-morph individual. Interestingly, I also observed changes in pollen length, which suggests that YUC6 might be involved in that pollen size dimorphism. To support the hypothesis that YUC6 is involved in auxin production, I also looked at the expression of a series of auxin-related genes that I had previously identified as differentially expressed between the L-morph and S-morph individual. And I found that a series of them actually were differentially expressed between the mutant individual and the S-morph individual in a manner that was similar to that of the L-morph plant. So that was cool. And further supported the hypothesis that YUC6 is involved in auxin production. Okay, so now I have a few slides about distyly beyond the genetic basis for anyone who's interested in anything other than genetics. So this is overwhelming, I noticed. So one thing that is kind of like the black sheep of distyly studies is that what is the basis of self-incompatibility in the L-morph? Like what's going on in the L-morph? Nobody knows. I think that potentially it means that there is a underlying self-incompatibility system that distyly was built off of. And I think this is mildly supported by a series of different plants that show some degree of self-compatibility, as well as our my turnaround knockouts. If there was an underlying basis for self-incompatibility that distyly was evolved from, then you would anticipate that individuals who are selfed would produce less seed than when they were illegitimately outcrossed, right? Which is something that we actually do see. So with my YUC6 plants, for example, we see a much lower seed set when we have them self than when we do an illegitimate cross. Furthermore, in populations that show some degree of self-incompatibility, illegitimate outcrosses typically have higher seed set than selfing. And in some populations, you cannot self at all, but you can produce seeds during an illegitimate outcross. This is like a very unsupported hypothesis that's just going off of like dreams, I guess, but I think that would be cool one day to expand on. So distyly is actually considered a potential stepping stone to dioecious. So a dioecious plant, wow, I put the wrong picture there. Oops, sorry. I put the wrong picture. This was supposed to be dioecious. I'm so sorry. Corn is monoecious because on the plant we have, yeah. So dioecious is when we have true sexes within a plant. So we will have a male plant and a female plant, kind of like ginkgo, or like Populus trichocarpa would be an example, like cottonwood or cannabis. Yeah. So I'm really embarrassed by that. I'm so sorry. Anyways, so we actually have 15 different genera that do have distylous and dioecious plants, and 6 actually have evidence that dioeci evolved from distyly. In these 6 genera, distyly is considered an ancestral trait, and thus a plant would either have to lose distyly altogether, become a homostylous plant, and then evolve dioeci from that homostyle, which makes less sense than just going from a distylous plant to a dioecious plant. You would imagine this is also a great system for this because we have our stigma that's already high up and is going to be more reproductively successful than the S-morph stigma, hypothetically, and vice versa. That reciprocal orchogamy can already push towards dioeci. Okay, and then in terms of population genetics, the breakdown of heterostyly is pretty cool. So in several genres, we have the reversion of heterostylist population to a homostylist self-compatible population. And this is rather interesting because this is going to be happening on the edge of species boundaries. So you can ignore the colors. Those just refer to different chloroplast genes. Instead, focus in on these white, black, and gray circles. So the gray represents population, homostylist individuals within the population. So individuals that have lost distyly. White is our L-morph individuals, and S is our S-morph individuals. And we see that in the majority of these populations, while we see homostylist individuals, we'll also see a large number of L-morph individuals, but we'll lose S-morph individuals. And this is likely because so far it's been documented in Primrose that it's mutations within the S-morph individual that are resulting in these homostyles, not the L-morph individuals. So our S-morph individuals are losing typically their female mating type gene, that SIP, and converting into homostylist individuals, particularly on population boundaries. This is just interesting in terms of you evolve self-incompatibility to prevent genetic inbreeding depression, but then if there's only a couple of you left on a species boundary, right, you have to just start inbreeding with yourself to actually survive as a population. Yeah, okay, now I open it up to questions. Yeah, no problem. Yeah. Oh, so it kind of, Turner fell into my lap. So during my PhD, Joel Shore from York University, he asked my advisor if he would like to help him on the Turner project. I think Turner is a great species because it flowers every day. Primrose, you have about a 3-month window to collect floral tissue, which is bad if you work in plant reproduction, right? But every day, Turner flowers, new flowers, that's great. Big, beautiful flowers, right? Not like Primrose, which are tiny. It's about like at least 3 times the size. And if I ever go on sabbatical, I get to go to like Cuba, you know. I don't know why Joel Shore decided to do it, but I don't know. I like Turner. Yeah. Yeah.
Fig plant. Flowers are actually inside the fruit. They have this strange mutualism of the fig wasp.
Oh.
The fig flower, right?
No, I have not.
Okay, it's really strange.
If it has a flower, then it is an angiosperm. Yeah, everything with flowers is an angiosperm. Oh, that is weird but cool. Now I'm gonna have to go home and Google it and go like a Wikipedia deep dive.
Tell me more about this that you put in a style that made it grow longer with the cells.
So
Elongating right there, yeah. So how similar is that to an oxen that cells grow longer?
breast and steroids in general, or in terms,
I'm looking at that. They got really long yellow lines there.
Mhm.
And you also have a really long style. And so when you say that was exogenous treatment, that means you dipped it on there or you put it on?
So
Just liquid on there at 10 micromolar?
this work was done by Leonard's lab. So Leonard is like the Primula guy, I'm the Turner girl. But yeah, supposedly they injected the buds with the bracillinoid, and then like you wait for the buds to develop because these the differences in morphology occur throughout bud development. So you'd have to inject it early on during bud development to ensure that you'd actually end up with a phenotype if there's going to be one. But the idea behind it is that SIP itself is basically making it so breast steroids are not available for the cells that are trying to elongate. And by providing it with that breast steroid, then it's capable of producing those elongated cells. There's going to be some pathway that's involved related to breast steroid, but at that point, at this current time, that's unknown.
Experiment where they have oxygen, they auger and stick it on the stem. And so this is kind of harkens back to your opening with the interest in Darwin, if I'm not misinterpreting this too badly.
No, I think that makes sense.
Other questions? Anybody at home watching on Zoom want any ask any questions? Probably put it in the chat. Let's see anything in the chat. All right. Otherwise, thank you very much. Appreciate it.
Hopefully